about amphetamine action. So we think about amphetamine binding to the dopamine transporter, which is presynaptically on dopamine neurons. It serves as a substrate for the dopamine transporter, and it inhibits dopamine reuptake. It enters the cell and has been proposed to serve as a substrate for the VMAT, which it does. And in so doing, it inhibits vesicular transport. It's also thought to enter the vesicle and essentially act as a weak base and deplete the catecholamine or dopamine stores from within the vesicles, thus increasing the cytoplasmic concentrations of dopamine. It's also been proposed by a number of groups to modify in some way through perhaps a series of phosphorylation events the dopamine transporter, rendering it giving it greater propensity to actually go through an efflux mode and essentially release neurotransmitter and increase dopamine efflux. And so that's why we think of them as dopamine releasers. So I wanted to mention a little bit about what we know about the regulation of the dopamine transporter. And I think this is an important point because this will tell you where we got started within the story. So there are a whole variety of mechanisms that are known to regulate dopamine transport. They can be regulated by, it's been shown by a number of groups, and I really wasn't able to acknowledge everyone that's contributed to all of this work on the slide. But essentially, it's been shown that phosphorylation can alter surface expression of the dopamine transporter. It's regulated by efflux, it's regulated by CAM kinase 2, and a whole variety of kinases have really been implicated in regulation. In addition, it's been shown that scaffolding proteins play an important role in determining the activity of the carriers, because essentially they are what helps anchor the carriers to in their appropriate places at the surface, the membrane surface. And so a variety of anchoring proteins have been identified, PIC1, PIC5, and they may be involved in trafficking or surface localization of the carrier. In addition, a variety of direct interactors have been proposed. Cholesterol has more recently been shown to actually interact directly with the transporter. This is actually the 3D structure from X-ray crystal structure that was from Eric Rowe's lab, showing a cholesterol molecule binding within the membrane to the transporter and essentially regulating its activity. And we had shown years before that, in fact, the binding of cholesterol to the dopamine transporter rendered it to be more outward-facing and available to bind neurotransmitter in that form. So it increased cocaine binding. In addition, G-protein beta-gamma subunits have recently been shown to modulate and inhibit directly the function of the transporter by binding to probably a C-terminal domain within the carrier. And again, this is another mechanism for direct interaction. So what we became interested in was a particular phenomena described in a number of labs, Jonathan Javich's lab, Randy Blakely's lab, and Ulrich Gether's lab, as well as Aurelia Valley, essentially had shown that substrates themselves have the ability to regulate the surface expression of the dopamine transporter. So methamphetamine can decrease surface expression, amphetamine can decrease surface expression, as well as dopamine. And so in many cases, these studies were done in transfected cell lines, which lacked the ability to synthesize dopamine or even release it. And so this began to suggest that these compounds, in some way, could modulate directly the surface trafficking of the dopamine transporter. And we became very interested in the mechanism. And so this just illustrates the phenomena using two movies, one of which, this is using turf microscopy, which sort of selectively illuminates the interface between the glass covers the aqueous phase, and essentially what you'll see when and these reflect internalization events. The dopamine transporter is fluorescently labeled in these cells, and what you'll see is that when you apply amphetamine, which we've shown on the right, you'll see a rather dramatic increase relative to the control of these puncta, these events, which involve internalization of the dopamine transporter. And that's just quantified here, where just counting the number of puncta, amphetamine induces a rather dramatic increase in internalization of cell surface stack. So we began to explore the mechanisms, cellular mechanism for this, by rounding up the usual suspects with respect to endocytosis. An obvious place to look would be clathrin dependent. And the next slide just simply shows that if you look at fluorescently labeled clathrin after amphetamine treatment and try to co-localize it with the dopamine transporter, and actually at any step along the pathway, we were never able to see co-localization. As you'll see, the red and green are quite distinct. And it seemed as though these studies really suggested that unlike transferrin, which undergoes clathrin-mediated endocytosis, which co-localizes quite nicely with the fluorescently labeled clathrin, the dopamine transporter, in the presence of amphetamine, does not appear to undergo clathrin-mediated internalization. So we then we looked at caveolin and flotilin dependent, but really saw no evidence for that. So that we then turned to another pathway for endocytosis of membrane proteins from the surface, and that's a clathrin and caveolin independent pathway that's dependent on dynamin, but also on a small GTPase within the cytoplasm, row A. And so, in order to explore this, we used a botulinum toxin that selectively inactivates row A, and we were able to show, in this slide, in a co-transfected cell with DAT and C3 exotoxin both being expressed, we saw essentially no internalization of the dopamine transporter in the presence of C3. But in cells which were only transfected with the dopamine transporter, we still saw robust internalization, supporting the idea that it was row A dependent. The dominant negative row A did not essentially block the internalization mechanism, again, as did C3. We looked further into it because, like other row-mediated events, it turns out that row-mediated events are often sensitive to inhibitors of actin dynamics. And again, the process fit with this in that blood-trunculin A as well as jesplakinolide, both which alter the dynamics of the actin cytoskeleton, essentially blocked the effects that we saw. So this was really strong evidence that in a transfected cell line, that the internalization mechanism of the dopamine transporter was row A dependent. So we began to explore further whether these events also occurred within the brain. And so we looked into two distinct systems. We used primary midbrain neurons and acute brain slices. And we developed a system based on some work by Alice Ting and her colleagues, where we essentially tagged an extracellular epitope of the dopamine transporter with what is referred to as an acceptor peptide, which is a domain that can be selectively biotinylated by an E. coli biotin ligase. We then co-expressed this biotin ligase, but included on it an endoplasmic ER retention signal, which would enable the biotin ligase to reside within the endoplasmic reticulum and essentially biotinylate the transporter as it goes to the surface. And once on the surface, this would enable us to use a variety of avidin-based reagents to really mark simply the surface-expressed carriers. And so doing this allowed us to explore this mechanism within dopamine neurons, and we can essentially show a lot of the same things. These are in Th-positive dopamine neurons, and in this case, we can show that the labeling at the surface is dramatically decreased by amphetamine. And I think the most surprising and curious finding in this case was that cocaine, another psychostimulant, which isn't a substrate for the dopamine transporter, blocks this effect. And so here provides sort of the initial evidence in our hands that these events required amphetamine to enter the cell to trigger these events. We went on to show at the EM level that that is actually associated with a population of vesicular bodies that are tubulovesicular bodies. They're not clathrine containing, so this is using an Adhd affinity reagent at the surface of a neuron, and then allowing internalization to occur. And what we saw was that the gold particles essentially were present on multivesicular bodies and tubulovesicular bodies, but not on clathrine-coated vesicles, as shown here. Again, tubulovesicular bodies are associated much more strongly with RhoA-dependent internalization. And quite curiously, if we looked back at some of the early papers localizing the dopamine transporter within the brain from Virginia Pickles' lab, the dopamine transporter was noted to be present very commonly on a population of membrane department that was akin to a tubulovesicular body. So we then wanted to ask the question whether we could actually see activated Rho, because this particular mechanism for endocytosis requires activation of Rho. And so we used an affinity-based peptides that were GST fusion proteins fused to a binding domain for activated Rho, and we used one for activated RAF as well, and also another for CD42, and looked for those. And what we were able to see is that if we looked in the brain, and these are in acute brain slices, we could see a very nice activation of Rho, and as well activation of RAF following the application of amphetamine. In addition, as I mentioned in the last slide, cocaine blocked these effects, indicating that amphetamine needed to enter the cell to have its effects. So we then use these affinity GST fusion proteins to actually try to localize where in the brain Rho is being activated. And I don't have to go through the entire slide, but this simply shows the TH staining within this section. So the affinity GST rotecan was overlaid on either a vehicle-treated or an amphetamine-treated slice. And what you can see is that the major difference between the control and the amphetamine-treated is a region of cell bodies localized within, that really quite nicely co-localized. This is simply the next section over, with the tyrosine hydroxylase positive, suggesting that the Rho activation we are observing in the brain occurs within these, within this population of neurons. And Rho activation, when we look closely, appears more prominent in processes than in cell bodies, and I'll get back to that a bit later. And so we then turn to another feature, which was actually well known. Amphetamine also increases cyclic AMP production. And this simply shows studies whereby you can apply amphetamine to, in this case, SK and SH cells, and show a rather nice increase in cyclic AMP. And the reason why we were intrigued by this is because we knew that amphetamine was clearly activating Rho once it entered the cell. But now the question was, if amphetamine also activates cyclic AMP, it's actually well known that activation of pKa is part of a pathway that inactivates Rho. And if you'll remember the time course I showed you within the brain, and I guess I could go back to it, but it was an extremely fast on, and within about 15 minutes had dropped down. And so we began to think that the interplay between these two pathways would be setting a window for activation of Rho and internalization of dopamine transporter. And so we went on to show that amphetamine-induced Rho signaling is indeed terminated by the pKa-dependent phosphorylation. And in this case, we used a mutant Rho-A that couldn't be inactivated. And you can see there's a nice potentiation of internalization as if Rho remains on longer. And that in the case of Rho-A, you can see that internalization, if you co-transfect that, is reduced. And so inhibitors of cyclic AMP, or increasing cyclic AMP, or pKa activity, as you would predict from this scheme, actually blocks internalization, as shown here, dibutyl cyclic AMP, and inhibiting pKa potentiates, suggesting that this wing of the pathway is open and is actually regulating the effects we observe. So we then turn to ask whether the activation of endogenous G sub S coupled GPCRs could prevent amphetamine's effects. And in this case, what we found was that SKNSH cells, which very nicely have a whole series of GPCRs that couple three different pathways, we could essentially block internalization of the amphetamine-dependent internalization by activating G sub S. And similarly, we could also, using a D1, D5 agonist, we could essentially prevent, at least partly prevent the internalization that we observed in neurons. So I'm going to bring you to sort of a summary slide, the first of several, that really illustrates what we think might be happening. Dopamine enters the cell, and, amphetamine enters the cell and results in the activation of Rho-A, which then in turn leads to internalization of the dopamine transporter. We also showed that it was dependent on phospholipase B2, which is actually fairly common for Rho-dependent internalization. And in addition, we could show that pathways that activate pKa in turn inactivate Rho and limit this process, and in fact, essentially determine the window over which the internalization appears. So we then asked whether we could test this or examine it behaviorally. And in this case, what we were doing was a very simple experiment where animals were treated with either amphetamine or cocaine. And we also, essentially, to activate the pKa pathway, used SKF 38393 to activate D1, D5 receptors. And this is, and this simply shows locomotor activity or ambulation, and in this case, we saw similar results with rearing, and that is that amphetamine increases the locomotor activity, and it's best seen here. Cocaine does as well, but that activation of the pKa pathway, which we've shown inhibits the activation of Rho, essentially blocks the increase in ambulation and locomotor activity in the presence of amphetamine, but it has no effect on cocaine, which is not transported into the cell. We could show that that was indeed internalized after the IG drug treatment, and so, yeah.